plugged in or?
Yeah. All right. Well, it's great to see everyone here this morning. I'm actually very happy to see this event come together. I'm going to call it the Meeting of the Minds because somebody used that in some nice talk, and I thought it was appropriate. So I would also like to thank Rob and Ian for organizing this event, and I'd also like to thank our administrators who helped make everything seem so easy and so smooth with getting this started. So you heard from Rob and Ian about their charge to all the speakers. So I looked at this as well. And I'm going to make some comments today about the Cancer Center here at Brown and some of the things we need to be thinking about as we do our research here, what some of the great opportunities are. And I'm also looking forward to hearing some cool stories throughout the day and thinking about collaboration opportunities. And I think it really is important for all of us and our leaders to be thinking about how to support interdisciplinary collaborative research here, because that really goes to the sum is greater than the parts and ultimately impact on the field. So over the last 6 years, we have been organizing a world-class cancer center called the Legoretta Cancer Center, whose vision is a cancer-free Rhode Island and beyond. Our mission is to be a world-class cancer program that brings scientific innovation, clinical and population research to reduce the burden of cancer in Rhode Island communities. We're committed to education, training, the next generation, and diversity in all that we do. Rhode Island has among the highest cancer rates in the country. And so as we talk about science and the opportunities, it's very important to reflect upon our community. The high cancer rates in Rhode Island go across cancer types. And for those of you who have lived here or know about the history here, there's a history of industry that goes back to the Industrial Revolution. And this history may have some impact on what we're left with today. And we also have, well, the other thing I'll say is as you go around Rhode Island, the high cancer rates are different in different parts for different types of cancer. So it's not like we have a hot spot where everything is high. There's more to it. It's more complicated. All right. It's important to remember that we have different races and ethnic groups in the state of Rhode Island. So this is Providence County up here, which has a large Hispanic population. And interestingly, among this large Hispanic population, there are multiple different races, which is important to be aware of. There are also quite a few different ancestries here in Rhode Island, and you could look at, you know, the African, Hispanic, and Asian, and where their origins are. So as far as the high cancer rates, I'll point out a few things. Rhode Island has a lot of toxic waste. There are 13 Superfund sites throughout the state with a history going back, well known. Brown had a Superfund grant in the past, and you know, it's important to keep talking about how we get back to addressing some things, but the state has a lot of data as well. And this is some data from the state and national information. As you know, radon, for example, is associated with lung and other cancers, and 1 in 4 homes in Rhode Island contain radon above EPA levels. Ultrafine particles are part of air pollution. They're found around airports and contribute to multiple causes of cancer. We are on the high side of these things as far as other parts of the country. And it was interesting to see the relationship between dose and cancer risk. It's not linear for some cancer types. For others, it is. And, you know, there's a literature about that for those who are interested. PFAS, also known as everywhere chemicals, come from textile and other industries that certainly have their history here in Rhode Island, and they are here in different distributions around the state. So some of the key areas of progress over the last 6 years are shown here as we have put together our cancer center. And I would like to highlight the Legaretta gift in 2021, our recruitment of our Associate Director for Administration, Basic Research, and Translational Oncology by the end of 22. And we've been busy, and I'm not going to walk you through all of this stuff. And we've especially been busy this year. And as I think about that, I think it's because of all these great leaders that we've recruited that are doing a lot of stuff, including having this cancer biology and bioengineering retreat. And, you know, obviously there are other important things. And in the audience, for example, our Associate Director for DEI, Sheldon Holder over there, and our Associate Director for Cancer Research, Training and Education component or coordination, Michelle Dawson. Our Cancer Center has 3 research programs in cancer biology, cancer therapeutics, and population sciences. And these are the program leaders. These are the themes, and I think as you consider ways of bridging between cancer biology and bioengineering, it's useful to know something about the strengths within the cancer research areas, and strengths in aging, inflammation, signaling networks, DNA repair, environmental carcinogenesis, stem cells, tumor microenvironment, and chemoresistance. And we are strong in translating knowledge. So a lot of what we do with our technologies is develop new diagnostic tools or new therapeutics or ways of delivering therapies or imaging. And ultimately, our goal is to apply this in the clinic to have impact. And in our population sciences, we have a very strong group that focuses on computational and informatics capabilities, and there are also groups that focus on behavior modification, because if you listen to the American Cancer Society, about half of all cancers are preventable if we just follow certain practices in our habits. The Cancer Center has shared resources that offer capabilities and technologies, and basically the goal here is to have innovation in the technologies to accelerate research and to promote collaboration. And one of the areas is in organoids that I think certainly applies to bioengineering as far as the next level of innovation. And here I'm talking about patient as well as transgenic organoids with control of alleles. And you can do a lot with these things, and I think they're going to be around, and they're already having application in the clinic. So as Rob mentioned, there's cancer engineering is a hot area, and there are a couple of recent manuscripts in Cancer Cell that are talking about this and providing conceptual frameworks for this emerging subfield. And the progress in cancer has been challenged. So what this shows is a bunch of different challenges that clinicians face as they treat cancer. Our drugs have toxicity, there's tumor heterogeneity, we don't have good models, and the tumor microenvironment and metastases are very, very complicated. And there are technological capabilities that are directly relevant to engineering and bioengineering that can be applied to this complexity. And I think we look forward to hearing about where we stand at Brown and what are some of the opportunities as the day goes by. Here's another one that talks about focusing on cancer. Just as you all know, there are hallmarks of cancer. There are hallmarks of or approaches that target these hallmarks from engineering, from the field of cancer engineering, that are opportunities for how there could be collaboration between the different fields. And as you can see in this schematic, it also involves the clinicians who will translate discoveries. So I just wanted to briefly show this, which is our rich portfolio of clinical trials at Brown's Cancer Center, from immune therapeutics to DNA repair to CAR T cells and various small molecules that are in translation. And so there is an opportunity to impact there, and I'll elaborate on that shortly. We also have a infrastructure to impact on translational research. And part of this infrastructure involves 8 translational research disease groups and leaders, including Sendai Mani, our Associate Director for Translational Oncology, and Attila Seyhan, who's sitting there, who directs translational oncology operations. And this is a busy slide, but this is essentially how the Cancer Center facilitates translation. So ultimately, what we want is value added and impact on patients and the community. And in the process of translation, this happens through novel clinical protocols that start with ideas and concepts. And so within the Cancer Center, there are all these different venues where you have ideas and opportunities being talked about and cooking. And then we have these translational disease groups that I spoke about. And these people talk to these people and facilitate the progression that you see here. This is supported by pilot funds that have specific focus of interest. And there's also an opportunity, or in the process, these meetings spin off working groups, which is something that Rob mentioned earlier, with certain deliverables, such as new protocols, new grants, and new collaborations that benefit from this sort of a infrastructure. So there's a huge opportunity, and I'm going to wrap it up. But I should tell you, I would have been done by now, except Ian Wong wanted me to talk about science. So I'm going to just show a couple of slides. Our own lab discovered has been working on this innate immune pathway for over 20 years. And along the way, we became interested in drug discovery. And so we discovered a compound called ONC201 that eventually went to the clinic and has been shrinking diffuse gliomas in the brain. And this trail pathway, just because it's near and dear, I just want to show you how powerful it is. The trail cytokine kills tumor cells selectively and specifically, and this is what lung cancer cells are doing over 4 hours when exposed to trail. This is a clinical trial that was published this summer about the efficacy of ONC201 in patients with brain tumors, and about 2/3 of patients have disease control, and about 1/3 of patients have responses, some of which are durable. Our own translational efforts here, led by Lan Zhu, is in the audience. I mean, I don't know if you could see this, but these are mice that are getting different treatments in different cohorts. And the longer these lines go this way, this is the better part of a year going down that way. And we're testing different drug combinations that are supporting future clinical trials with these novel compounds. I wanted to mention something about pneumonitis, because in cancer, we care about toxicities and we want to prevent toxicities. It turns out that the trail pathway suppresses radiation pneumonitis, and we're now looking at immune checkpoint pneumonitis, and the phenotypes are quite impressive, and our genetic knockouts are particularly susceptible. This is recent progress. I don't know if Jill is sitting there somewhere, but we've recently been able to show CCL22 can also suppress this phenotype. And these are 5 different mice in each treatment cohort. The ones at the bottom are the ones that are rescued. And I'd love to just hear from the audience. Do you think there's evidence of rescue? Because Jill is not convinced, but we're trying to quantify this and do more with it. But to my eyes, it certainly looks like there is a phenotype there. But the lungs are complicated, and we do need an organ on a chip. And it's complicated further by all the different cells that need to be there, and even things you can add to it. We are collaborating with Teresa on a microRNA we discovered from our hypoxia work, and we are looking forward to having ways of delivering it and testing it in vivo. And we do make organoids, and I didn't know that organoids did this, but they dance, okay? They even do somersaults, which is amazing that they do this. And I wonder how different microenvironments can affect this behavior, and whether we can do anything to exploit these types of biological behaviors that are just not so well known. The last thing I'll show you is an opportunity in precision oncology. So clinicians want to treat patients better with newer and better ways, and what's the best we can do for a patient in 2024. And as we design clinical trials to do this, there is an important part that technology gets to play in the context of these clinical trials. And technologies have an opportunity to be validated with patient samples and clinical outcomes. Okay, and so I would encourage you, because we do have leadership in this consortium, and I think RNA modifications are low-hanging fruit here, but there are definitely other platforms that could have clinical impacts. So in summary, I think we need good models that reflect the patient's tumor microenvironment. And it's very, very important. And the models need to have context, such as, are patients newly diagnosed? Are they at recurrence? Are they resistance? Our models need to be informed by the racial and ethnic diversity within our community. Our models need to be dynamic. So we need serial imaging at high resolution. And I don't have time, but obviously there are a lot of different cell types to image and study. We should take advantage of the translational infrastructure and take advantage of institutional and cancer center pilot opportunities. So I'm looking forward to hearing some cool stories. And I just want to give a plug for some upcoming retreats, the Translational Oncology Retreat and the Cancer Center Membership Retreat. So thank you very much.
Thanks, Rafiq. Always a great start to our meetings. Appreciate that. And now I'm going to just